Nucleic acid structure. DNA and RNA are large macromolecules with several levels of complexity. Nucleotides form the repeating units. Phosphodiester bonds link nucleotides to form a strand. Two strands interact to form a double helix. The double helix interacts with proteins, resulting in 3D structures in the form of chromatin. 3D structure, nucleotide components. Figure 9.8. 9 to 25. Copyright the McGraw-Hill Companies Incorporated. Permission required for reproduction or display, combining all the parts. Base plus sugar nucleoside. Example: adenine plus ribose equals adenosine. Adenine plus deoxyribose equals deoxyadenosine. Base plus sugar plus phosphates nucleotide. Adenosine monophosphate, AMP. Adenosine diphosphate, ADP. Adenosine triphosphate, ATP. 9 to 27. Copyright the McGraw-Hill Companies Incorporated. Permission required for reproduction or display. 9 to 28. Figure 9.10. Base always attached here. Phosphates are attached there. Figure 9.10. 9 to 28. Figure 10 to 08. Caption: Structures and names of the nucleosides and nucleotides of RNA and DNA. Figure 10 to 09. Caption: Basic structures of nucleoside diphosphates and triphosphates, as illustrated by thymidine diphosphate and adenosine triphosphate. DNMP, NMP, A, G, C or T, A, G, C or U. Figure 9.9. The structure of nucleotides found in A, DNA and B, RNA. 9 to 26. Nucleotide polymerization reaction, phosphodiester bond formation. Figure 11 to 08. Caption: Demonstration of 5 to 3 synthesis of DNA. Figure 9.11. 9 to 30. Copyright the McGraw-Hill Companies Incorporated. Permission required for reproduction or display. Events leading to DNA structure determination. 1953, James Watson and Francis Crick discovered the double helical structure of DNA. The scientific framework for their breakthrough was provided by other scientists including Linus Pauling, Rosalind Franklin, Erwin Chargaff. 9 to 31. Copyright the McGraw-Hill Companies Incorporated. Permission required for reproduction or display. Linus Pauling, Rosalind Franklin. Helical, double-stranded, 10 base pairs per turn. X-ray diffraction pattern of DNA. Erwin Chargaff's experiment. It was assumed the 4 bases, A, G, C and T were in a repeating, tetranucleotide configuration. Therefore, there should be the same amount of A, G, C and T in any molecule of DNA for any source. Chargaff carefully determined the exact percentages of nucleotides in DNA from several sources. 9 to 35. Copyright the McGraw-Hill Companies Incorporated. Permission required for reproduction or display. Erwin Chargaff's data %2 equals %T and %G equals %C. However, %T did not equals %GC. This observation became known as Chargaff's rule. 9 to 39. Watson and Crick modeled DNA structure. Base pairing key to DNA structure. Features of the DNA double helix. NH 2 T 2 nanometers 1 nucleotide. 0.34 nanometers. 1 complete. Turn 3.4 nanometers. O H N P O NH2 O H2N Ho 5 cents end 3 cents end 3 cents hydroxyl 5 cent phosphate A S C G CH2 CH3. Features of the DNA double helix. Minor groove, minor groove, major groove, major groove. Ball and stick model of DNA. Space filling model of DNA. Major helical conformations of DNA. A DNA. bDNA. Melting point curve, trademark is proportional to %GC. Figure 10 to 14. Caption. Increase in UV absorbance and temperature, the hyperchromic effect, for two DNA molecules with different G triple bond C contents. The molecule with a melting point, trademark, of 83 degrees Celsius has a greater G triple bond C content than the molecule with a trademark of 77 degrees Celsius. Trademark equals 68.9 plus 0.41% GC, the three-dimensional structure of DNA. Figure 9.21. 9 to 55. RNA structure. The primary structure of an RNA strand is much like that of a DNA strand. RNA is made as a single strand only, however it may form a double-stranded structures. RNA strands can be 10 to 1000 s of nt in length. RNA is made from a DNA template, only one of the two strands of a DNA helix is used as the template. RNA contains uracil rather than thymine. 9 to 57. Figure 9.22. 9 to 58. RNA secondary structures. Figure 9.23. 9 to 60. RNA tertiary structure, atrana. Figure 9.24. 9 to 61.